To understand first how semiconducting nanowires can serve as chemical or electrical sensors, we have to understand the physics of the nanowires that are contacted between two electrodes and integrated in a platform of field effect transistor. For the sake of clarity, I will focus on the cases of silicon nanowires. In the presented diagram, a p-type nanowire, which seen in the upper image, and n-type nanowire, which seen in the lower image, are contacting at both ends to metal electrodes. As for a conventional metal semiconductor interface, the silicon nanowire bands bend up for p-type silicon nanowires and bend down for n-type silicon nanowires to bring the nanowire Fermi level in line with that of the metal contacts. When the gate voltage in the field effect transistor is positive, then the bands are lowered. This action actually depletes the holes in p-type silicon nanowires and as a result suppresses the conductivity of the silicon nanowires. For the n-type silicon nanowires, this lowering of the bending bands leads to an accumulation of electrons in the n-type silicon nanowires and therefore enhances the conductivity. Conversely, when the gate voltage is bands are raised and as a result the conductivity is increased in the case of p-type silicon nanowires and decreased in the case of n-type nanowires. Optical doping of the silicon nanowires can lead to improved properties of such devices, and the ability to design these properties according to the desired applications can be well achieved. A selective sensor can, in general, be configured from silicon nanowire device by linking recognition receptor groups to the surface of the nanowire. A typical procedure for the preparation of molecularly modified silicon nanowire sensors include: first, attaching a molecular film to the silicon nanowire surface. In case the silicon nanowire is coated with oxide layer, then silane-based molecules are used as seen in the presented example on the screen. In case the silicon nanowire is not coated with oxide layer, then the modifying molecules in the case of silicon could be attached throughout silicon-carbon bond. In few instances, the molecular layer can be used as intermediator to connect other functional groups that organically cannot stick or bond directly to the silicon nanowire surface. In the presented case on the screen, the aldehyde silane molecules on the surface are used as a mediator to connect antibodies that shall serve for sensing the targeted molecules in a liquid phase. And in the last step, the targeted molecules are attached to the molecular modification and the sensing process is therefore accomplished. The sensing process itself can be configured from field effect transistor device as illustrated in the presented schematics. The target molecule in this case, such as protein, usually have a net positive or negative charge in the aqueous solution. In this case, specific binding will lead to a change in the surface charge and a change in the nanowire device. By monitoring the conductance of the nanowire device, the detection of the target molecule can be achieved. A more specific example on the sensing process of the pH by silicon nanowire field effect transistor is presented in the current slide. Here, a silicon nanowire field effect transistor is formed by modifying the silicon oxide surface with a 3-amino propyl triethoxysilane. And in this case, the modification provides a surface that can undergo protonation and deprotonation. Changes in the surface charge due to these reactions change the electrical properties of the nanowire. Measurements of the conductance as a function of time and solution of pH demonstrates that the nanowire conductance increases stepwise with discrete changes in pH from 2 to 9. And this can be seen in figure number B on the screen. In addition, the conductance is constant for a given pH. A typical plot of the conductance versus pH shows that this pH dependence is linear over the pH that range between 2 and 9, and this could of course seen in figure or panel number C on the screen. Actually, the observed linear response can be attributed to an approximately linear change in the total surface charge density, and in this case, to support this point, pH-dependent measurements on unmodified silicon nanowires were performed. This could be seen in figure D on the screen. In this case, these conductance measurements show a non-linear pH dependence. Indeed, the conductance change is small at low pH, namely between 2 to 6, but large at high pH range, that is usually range between 6 to 9. The conclusion from these experiments is that modified silicon nanowires could function as nanoscale pH sensors. Here is an animation of real detection by silicon nanowire field effect transistor. And as seen in the video, the conductivity of the nano sensors increased dramatically when the molecules adsorb at the chemically modified surface of the nanowire. When the concentration of the molecules is doubled, so is the sensor signal. In general, the change in the conductivity is proportional to the molecule's concentration. This animation could be appropriate actually for a silicon nanowire array that is used for detection of specific biological markers typically present in cancer patients. The science behind these sensors is that an antigen in the sample binds to its corresponding antibody on the nanowire. The antigens are slightly charged, so when it adheres to the antibody, an electric field is introduced to the underlying silicon nanowire. Charges inside the wire redistribute to cancel out the electric field, resulting in a change in the wire's conductivity. This change can easily be measured electronically, and the antigen concentration can then be determined. This electrostatic effect is the foundation for modern field effect transistor and can only be observed at the nanoscale. Despite this great promising of these devices, there are still many problems that must be solved before fully integrated device can be realized for early warning of cancer detection. I have to mention that this FIT-based sensing approach has been extended to hundreds of various target molecules and applications. Nevertheless, the principle is more or less similar, and therefore I would stop at this point and move to the other topic. A promising silicon nanowire-based sensors for in vivo or cellular applications was recently developed and tested. Researchers have demonstrated that necked nanowire structures in which the active field effect transistor channels are encoded at or close to the neck tip can be synthesized in a straightforward and high-yield manner. Probes that are synthesized with 60-degree tip angle and formed with two cis-linked 120-degree necked units and represent an ideal geometry for intercellular insertion, although a single 120-degree neck can also yield a viable probe. In these nanostructures, doping modulation are used to introduce a short channel field effect transistor topography defined very closely to the probe tip, and heavily doped nanowire arms directly wire up the field effect transistor channels as seamless nanoscale electrodes. This suggests that insertion of the neck tip into the cell could readily lead to the detection of the intracellular potential variation versus time in this case. Actually, using this device architecture, the researchers have shown that the probes can be used to measure changes in electrical potential outside, inside, and across cell membranes, and pH changes. More details on this approach could be achieved throughout the link seen on the screen.